Hello everyone, I'm Alex from CatchGene. Very glad to be here today. In my part, I'm going to introduce automated capturing of cell-free, circulating cell-free DNA, FFPE tissue DNA, and stool DNA. Okay, let's move to the first topic of my presentation, rare and small circulating cell-free DNA. Okay, about circulating cell-free DNA. Everyone have circulating cell-free DNA in our body. Here is the normal cell. Due to apoptosis or necrosis, DNA inside of the cell will release into our circulating system. So we call it circulating cell-free DNA. In 1 milliliter whole blood, normally the amount of cfDNA only 1 to 20 nanogram, which is only 1 per 10,000 of genomic DNA. And the size of cfDNA actually very small, only 150 to 200 base pair, which is only 1 per 1,000 of genomic DNA. And if there are some tumor cells inside of our body, the tumor cell itself will also apoptosis or necrosis. The DNA inside of the tumor cell will also release into our circulating system. So we call it ctDNA, circulating tumor DNA. And ctDNA actually also kind of cfDNA, but it's only in around 0.5 to 5% of cfDNA. And in before, people think this kind of circulating cell-free DNA actually a kind of recycle of the DNA. But recently, due to the detection sensitivity is higher, NGS digital PCR is more popular, and more DNA label biomarker. Now we know if we can capture those circulating cell-free DNA, then we can detect if there are any cancer cell inside of our body to do cancer early detection or monitoring. Okay, and actually there are lots of different sources for liquid biopsy. Majorly is blood, but also we got ctDNA from cerebrospinal fluid, from saliva, also from urine. And there are many different applications of liquid biopsy. First is we can use it to do molecular profiling to select treatment. We call it companion diagnosis. Also, we can use liquid biopsy, use ctDNA to do minimal residual disease detection. And also we can use it for cancer screen and cancer early detection. Okay, but if we want to do follow the applications which we mentioned in previously, the first of all, what we need to do is to purify cfDNA. And actually, there are many challenges to purify cfDNA. For example, first, it's very rare. As we mentioned, it's only 1 to 20 nanogram in 1 milliliter whole blood. And second, it's very small. It's only 150 to 200 base pairs. So it's very rare and very small, so not very easy to be captured. Currently, for automated extraction system, most of them still based on magnetic beads method. Actually, this kind of technology already exists for 15 to 20 years. People use magnetic beads. The red circle is red magnetic beads coating with silica to capture genomic DNA for genetic analysis or to capture viral nucleic acid for disease detection. Normally, the magnetic bead system handle only 200 microliter to capture very rich nucleic acid. But nowadays, what we want to capture is very rare cfDNA, cfRNA for precision medicine. So normally the scientists suggest to enlarge the sample volume from 200 to 4,000 microliter. If we still use magnetic beads, and it's become kind of very difficult to let the beads to capture the DNA. So actually, magnetic beads system are not good for rare nucleic acid. So if there any solution for this kind of rare nucleic acid, yes, here we are. Here, eye catcher was born for liquid biopsy. We have features like eye catcher is a membrane column-based system, like fission net, to capture more rare nucleic acid. Also, we use wash method. This is kind of method like take a shower to remove all inhibitors away. And also, we can handle large volume input. Uh, we can handle up to 4,000 microliter, which is 20 times than normal extractors. And also, we handle the sample, one sample in one line. So it can eliminate the possibility of cross-contamination. Okay, so how to capture rare and small cfDNA, as we mentioned a little bit. Other systems, they use magnetic beads like fishing hook. So only the beads touch or hook the DNA can be recovered. And in our eye catcher system, we use a special patent technology. We call it HPMC, high pressure membrane capture technology. It's kind of technology like fishing net. So we can recover over 90% of circulating cell-free DNA back, like a fishing net. And this is our kit. We can, the name is Circulating cfDNA 4000 kit. And we can capture the cfDNA from different donors and like high cfDNA level donor, medium or low cfDNA yield donor. And no matter in high, medium, or low, the consistency also show, both of them shows very good inconsistency. And we also did parallel comparison between eye catcher and the Q-brand S system, which actually, I show the full name here, which is Symphony system. We do parallel comparison, 4 milliliter whole blood and 4 milliliter plasma sample and parallel purify by eye catcher and Symphony. And after purification, we use QSEP1. We run capillary electrophoresis to see the peak of cfDNA and compare. And from this parallel comparison, we can see eye catcher can capture very high yield of cfDNA, much higher than Symphony system. If we see the RFU, which means the intensity of the cfDNA peak, the average of the intensity of eye catcher is 3.53, and Symphony is only 0.98, which shows 4 times higher cfDNA than Symphony system. Okay, and in Taiwan, we have a reference lab. Their name is ACT Genomics. In before, they use Symphony system. And now, after they have some problem, the cfDNA recovery rate is too low and not easy to do library construction. And they keep searching for different solutions. After they try eye catcher, they modify SOP and shift all their liquid biopsy sample from Symphony to eye catcher. And they use it actually for purified cfDNA from serum, plasma, or CSF, which is cerebrospinal fluid. And they use it for cancer monitoring. Okay, so that's the first part of my presentation, and let's move to the second part of my presentation about hazardous and fragmented FFPE tissue DNA. Okay, and about tissue biopsy, people know it's a golden standard of cancer diagnostic. If one doctor, they think the patient got some chance to have cancer. Normally, they ask the patient to have a surgery to collect the tissue, made it as FFPE and do slice, then made a slide to do the microscopy test to check if there are any tumor cell inside or if it's normal. And right now, from this microscopy sample, we can do DNA purification and to check the molecular profiling to select proper treatment for same kind of cancer. There are different subtypes. So for different subtypes, maybe have proper treatment for them. So we need to do molecular profiling. And this kind of molecular profiling, we call it companion diagnosis. And also for companion diagnosis based on FFP DNA, there are also two challenges. One, because FFPE, they need to deparaffinization, and normally people need to use xylene, and it's a hazardous chemical, and also the procedure is very complicated. It's a long procedure, so it's a hazardous and complicated treatment. And second, we all know FFP DNA, due to the production procedure, the FFP DNA usually already fragmented from 300 microliter to from 300 base pair to 1,000 base pair. But due to purification procedure, if we not take care the DNA, normally we will further destroy the DNA, fragmented DNA into smaller pieces, like less than 100 base pair. If this situation happens, it will be very difficult for following detection. So we need to avoid further destroy of fragmented DNA. So these are the two challenges of FFP DNA purification. About the first one challenge, how to symbolize the paraffinization step. Traditional, as we mentioned, we use lysine and it's very complicated. For right now, iCatcher, we develop a special reagent. We call it DWX, actually a dewaxing buffer. It's in yellow color like this. So we can symbolize the very complicated step into one step. We just need to do 60 degree, 20 minutes, and with a very safe, xylene-free, very safe dewaxing buffer, we can move the wax and let it can be DNA purification. So it's a very simple way to overcome the deparaffinization step by DWX buffer. Okay, and how to avoid further destroy of FFP DNA? We know magnetic bead system here is the way. During binding step, they use vigorous agitating to let the beads can mix, can touch the DNA. And this also kind of shearing force, which will cause the DNA damage, because if one DNA touch to two different magnetic beads, it's a shearing force, where that is degrade more seriously. And in iCatcher, we use a special, we use membrane. Let the lysate pass through the membrane gently, so we can keep the integrity of the FFP DNA in larger fragments. So this is a way we avoid further destroy of FFP DNA. And in iCatcher system, we got fully automated and semi-automated protocol for FFP tissue DNA. For fully automated, which is for normal tissue biopsy, just do dewaxing outside. And after dewaxing, they put all the steps into the system from lysis, binding, wash, dry, elute, all fully automated in the system. Only for some needle biopsy or for some very treacherous sample, if you want to get highest yield, you can do lysis outside until tissue all lysed. Then put it into eye catcher for also fully automated purification. And here is the data for FFPE DNA purified by eye catcher system. We can see we can keep the integrity of the FFPE DNA from 100 to over 5K, which is 5,000 base pair. And we can get very high purity over 2 and very good DNA quality. Okay, so that's the second part of my presentation, and now we move to the third part about difficult lysis and high inhibitor samples, which is stool DNA. Okay, stool DNA, actually, we purify stool DNA is to study gut microbiome analysis. We do microbiome analysis actually, and normally there are 4 steps to do the study. First, we need to sampling, then we need to purification, then we do quality control, then NGS analysis. Then we can get the bacterium population information, and we can use it to study many different aspects of studies. But also, if we want to do microbiome analysis, the very first step is to purify the stool DNA, and also challenges for stool DNA purification. The first one is we want to see the whole population. So no matter gram-positive and gram-negative bacterium, we need to lyse it. And we know it's very easy to lyse gram-negative bacteria, but it's very difficult to lyse gram-positive bacteria because there are so many layers of peptidoglycan. So we need to overcome the first challenge. The second challenge is easier to understand but difficult to solve it. Inside of the stool DNA, actually, there is super high inhibitors because stool sample is come from many different ingredients, and it's incubated in our stomach for a while to gut. So there are so many inhibitors inside. Okay, so how to complete lysis of gram-positive bacterium? Okay, our solution is we do actually 3 different aspects of solution. First, we do molecular. We use microbeads to do disruption. Second, we use stronger lysis buffer to do chemical lysis, to stronger chemical lysis. And third, we use physical solution, which is 95-degree treatment to let it lysis properly. So therefore, we can put it on the automation to do the rest following automation purification procedure. And another more difficult to solve problem is how to remove inhibitor and impurities by other automated system, by other magnetic bead system. Usually during washing step, they just like take a bath. They use magnet to absorb all the beads and move the beads from one bath to another bath. And you know, beads always in circular shape. So between beads and beads, there are some remaining buffer inside. So the remaining buffer from, for example, from binding buffer, very high salt and detergent will carry over to wash buffer, and then carry over to another wash buffer, and then finally to eluate. So here, the way the magnetic bead system to do the washing step is just to wash the inhibitors, just like take a bath. You know, everyone have the experience of take a bath, and if all of our body full of soaps, full of bubbles, even we jump lots of baths, there's still bubbles on our body. So it's not a good way to handle large volume or high inhibitor high impurity sample. And our solution for eye catcher, we use, because we base on membrane column, so we use a very special way. We use a wash away method, like take a shower to wash all inhibitor, to wash all impurities away. Here is the video. We do wash away again. We do several times of wash away to remove all the inhibitors, all the impurities away. So it's very efficient and won't carry over. So it's a very good way to handle stool sample. Okay, so here is a stool. Yeah, obviously. And in before, people only collect 1 point of the stool, maybe 0.1 gram stool sample, and preserve into 0.5 milliliter preservation buffer to do the 16S metagenomic sequencing to check the bacterium population inside of the stool. But nowadays, actually all the scientists not only interested in 1 point of the stool, they want to maximum sample input, because only 1 point of the stool may not represent the whole population. Maybe different position of this stool, the bacterium population is different. So they want to collect more part of the stool. So the sample become larger, not 0.1 gram anymore. It's become 2 gram and put into 4 milliliter preservation buffer. And they want to purify more bacterium DNA to do shotgun metagenomic sequence. Not based on this kind of sequencing, they can know more unknown bacterium, know more information about it. And this is also a very special solution. Actually, currently, as I know, no other purification system can handle such a large volume of lysate and very dirty stool sample. And eye catcher, we provide a eye catcher stool DNA 4000 kit, which can handle 4 milliliter preservation buffer. And we got very good purity and very good DNA quality from very dirty stool sample. Okay, and here is our reference lab in Taiwan. Their name is BiOptic. They are the biggest metagenomic analysis service company in Taiwan. So they handled around 10,000 stool samples annually. And before they use it manually, so they need to deal with stool every day, and everyone in their lab are unhappy. But recently, start from 2019, they introduced eye catcher system. So right now, they're very happy because they use fully automated system to handle stool and to eliminate the manual works. Okay, so here is a video to let us know more principle of our eye catcher systems. And here is our consumables. One sample are in one line, so totally we can handle 12 samples in one page. And it's touch screen, so it's very easy to handle the system. Okay, so start from lysis, it's fully automated. We can handle up to 4 milliliter sample, which means 12 milliliter lysate. And different from other systems, we use membrane column. We use membrane to binding. So therefore, we can get higher recovery rate, higher yield. And also during washing steps, we not use bath wash. We use shower wash to wash away all impurities. Okay, and after wash, we also eliminate ethanols by dry. And finally, you can elute in 30 microliter minimum, which is the lowest elution volume currently in the automation system, and get very pure DNA. Okay. Okay, here is CatchGene capture cfDNA, cfRNA, and microRNA automatically. Okay, so to summarize my part of presentation, iCatcher is born for liquid biopsy, and we can handle large volume from 250 to 4,000 microliter. We are membrane-based, we wash away. And actually, we not only can handle liquid biopsy, we also can purify normal genomic DNA from whole blood, also can purify RNA from blood or even from plant sample. We also can purify viral DNA, RNA from viral sample. Also, we can purify tissue biopsy and dirty and large volume sample like stool DNA or 4 milliliter whole blood sample. And the system, this is our system with a very compact footprint and can place it on the desktop. Okay, so this is my part of presentation.
Hello everyone, thank you to join us today. After Alex's impressive presentation, we will go into the second section of this webinar. In this section, I will introduce you a high efficiency and accurate quality control methods for various nucleic acids, including cell-free DNA, FFPE DNA, stool DNA, and total RNA. I'm Jamie from BiOptic. My presentation will divide into 3 parts. Firstly, I will introduce common DNA RNA quality tools in laboratories. Next, we will discuss the key factors of quality control for cell-free DNA, cfDNA, and how to perform the cfDNA QC with the high-efficiency approaches. Last, I will show you more QC applications on capillary gel electrophoresis CGE system. After isolating procedures, people will need to check the quality for their samples, including the purity, size, and the concentration, to evaluate whether the samples meet the requirements of downstream experiments. The most common quality control tools for DNA RNA qualification is electrophoresis. Electrophoresis is separating the materials based on the charge and the mobility under an electric field with electrolyte. The materials with the positive charge will move toward the cathode and negative charge will move toward the anode. The factors of effect molecular mobility will including the input voltage of electric field, molecular weight of separate materials, and the materials as electric point. Following are 3 common electrophoresis technologies. Gel electrophoresis is widely used in laboratories. The agarose polymerase will associate together to become a molecular sieving, and then separating the DNA according to their size, and using the property of the fluorescent dye to make DNA visible after specific light wavelength excitation. Another electrophoresis technology with a long history is capillary electrophoresis. Capillary electrophoresis is composed of three systems: capillary with the electrolyte, and the high voltage system and the high sensitivity optic system. The materials are separated in capillary filled with the electrolyte under the high voltage applied. When the DNA passes through the detector, it will be converted to a signal. Compared to the traditional slab gel, capillary electrophoresis is fully automatic. Because the DNA is separated in capillary with the 25 to 100 micrometer inner diameter, the sample consumption is low and the resolution is high. The most important is that the electrolyte can be changed to apply for different applications. In recent years, there is another popular technology, microfluidic chip. It narrowed down the long capillary into a small chip to minimize the sample requirement and decrease the analytic time severely. However, the cost of consumables is very high. We will briefly go through the advantage and the disadvantage of these three systems. Before analysis, users have to take at least 30 minutes to prepare the gel and do the manual loading for the slab gel and the microfluidic electrophoresis. But as we mentioned, the fast analytic time of the microfluidic chip is very fast, and the traditional CE with the high performance, with the high resolution and high sensitivity. Advantage of the slab gel is the cost is pretty low, so everyone can affordable the price. BiOptic wants to keep the advantage and improve the efficiency and quality of existing electrophoresis system for biofragment analysis. So we provide the users an innovative capillary gel electrophoresis, the QSAP biofragment analyzer. QSAP is an automatic system can process the 1 to 96 samples without gel preparation and the manual sample loading. And all analysis can be completed in few minutes with low sample consumption and good performance, high resolution, and high sensitivity. QSAP can resolve 1 to 4 BP and detect the 5 picogram per microliter samples. The most important is that QSAP can do the qualify and quantify samples for the DNA sample in single analysis without extra sample waste. The disposable pen-shaped cartridge design makes the analysis become easy on QSAP biofragment analyzers. The cartridge is composed of the capillary and the precast gel. Each cartridge can analyze 100 to 300 samples with fully automatic sample injection. Also, we apply more than 10 different precast gel into the cartridges to be adapted for different applications. Users can start the analysis in 3 steps to perform the QSAP platform. Insert the cartridge, place samples and buffers, and then click Run to get the results. During the analysis, the samples will be injected into the capillary by electrokinetic principle after apply the voltage. The DNA with the net charge will move from down to up and separated based on the molecular weight. When the DNA passes through the detection window, the system will transfer the signal to PIC diagram, and all procedures are automatic. This is the DNA later analyzed on QSAP system. We can see QSAP provides the accurate size information on the diagram directly, with the high sensitivity to detect the picogram level DNA and high resolution performance. And all analysis is completed in 3 to 5 minutes. Next, we will present how to do the quality control for various applications on QSAP system. The first one is the qualification and the quantification of the cell-free DNA on QSAP platform. There are two major points for self-DNA qualification: size and the purity. The self-DNA is from fragmented genomic DNA with the mixing size. The major size of the self-free DNA is about 150 to 200 base pairs. In some cases, there also exists diploid, even triploid, due to the duplicated core DNA. Since the most of cell-free DNA is isolated from body fluid, such as the blood or urine, genomic DNA contamination needs to be avoided. In addition, the carrier RNA can enrich the extracted amount of self-DNA during the isolation. So preventing carrier RNA contamination is one important QC point for the purity check. Following are the self-DNA samples which are analyzed on QSAP platforms. QSAP can identify the distribution of cell-free DNA in 3 minutes and distinguish the dinucleosome and the trinucleosome cfDNA. Next, in order to test the sensitivity of the QSAP platform for self-DNA detection, we serial dilute the cell-free DNA and test these samples by high-sensitivity cartridge, the N1 cartridge. The self-DNA stock concentration is detected by qubit. We can see that after 50 times dilution, the expected concentration of cell-free DNA is 4.2 picogram per microliter, and such low amount cell-free DNA is still detectable on QSAP platforms. Except the checking the size and distribution of cell-free DNA, QSAP can identify the purity of cell-free DNA when the samples contain other contamination, such as the carrier RNA. Sometimes, if the serine was under inappropriate preservation and the isolation procedures didn't remove the genomic DNA completely, we will see the contaminated genomic DNA display a fragmental pattern with different size, and we call it as the gDNA ladder. A QSAP platform can not only identify the contamination pattern of the cell-free DNA, but also provide the exact percentage of the target cfDNA. It could help the scientists to determine whether the quality of the cell-free DNA is adapted for downstream experiments or not. Here is an example. The cfDNA is prepared for next-generation sequencing, but we can see there exists about 80% genomic DNA combination, and it might interfere the following NGS library construction. cfDNA is rare. Cell-free DNA concentration is an important QC information except the size and purity. Here are the common quantitative tools in the market. We can divide these quantitative tools into two types. The first one is the detecting the concentration based on the wavelength absorption, such as the photometer and the fluorometer. Another type is the fragment analyzers. They detect sample concentration depending on other non-concentration DNA ladders, and QSAP is belong to this type. As mentioned, the quantitative principle of photometer and the fluorometer is based on absorption, so it means the materials with the same OD, like DNA and the RNA, would be detectable at the same time. Therefore, when we calculate the cell-free DNA concentration by photometer or fluorometer, it is overestimated easily due to the other materials combination, such as the genomic DNA and the carrier RNA combination. Furthermore, extra sample consumption is also a concern during the cfDNA qualification and the quantification. Here is an example. The cell-free DNA was extracted from plasma, but it contained the genomic DNA contamination. We stereo dilute the cell-free DNA and detect the samples by fluorometer and the QSAP at the same time. According to the results, the QSAP presents the high sensitivity and the high linear regression for the cell-free DNA quantification. QSAP can not only provide the size and the purity, but also accurate cell-free DNA concentration without other contamination interference in single analysis. Here are other examples. The concentration of sample 1 is about 2 nanogram per microliter detected by fluorometer. However, when we analyze this sample by QSAP platform, we can see that over than 90% is genomic DNA contamination, and it will affect the downstream experiment severely if we only do the quantification by fluorometer. The sample 2 is a result shows that QSAP system provides the accurate concentration for the mononucleosome, dinucleosome, and the trinucleosome cell-free DNA. Now, I would like to do a brief summary. QSAP is an easy-to-operate system. 3 steps can start the analysis and get the result in 200 seconds for cell-free DNA QC. In addition to the features of simple operation and rapid analysis, QSAP can detect low amount cell-free DNA due to the high sensitivity property. The most important is that QSAP can provide the accurate size, purity, and the concentration for cell-free DNA in one analysis. Therefore, QSAP biofragment analyzers are high-efficiency platforms for cell-free DNA quality control. After discussing the quality control of cell-free DNA by QSAP system, I would like to introduce you about more applications on QSAP, including the genomic DNA, DNA and RNA from the FFPE samples, genomic DNA from stool, and the total RNA quality control. Genomic DNA belongs to a high molecular weight DNA. It will display the smear pattern on agar shell. So scientists it's difficult to get the accurate size information when the genomic DNA is partially degraded, especially. After analyzing the genomic DNA on QSAP platform, QSAP can automatically calculate a village size of the genomic DNA as well as provide the DNA quality number, the DQN number, as the integrity index for the genomic DNA quality control. The DQN number is from 1 to 10. When the genomic DNA is degraded, the DQN will be lower as the sample 2 compared to intact genomic DNA. In some downstream experiments, such as the real-time PCR, partially degraded genomic DNA is still workable for the following gene detection. So, QSAP can provide the percentage of different regions in the genomic DNA sample. It would help researchers to evaluate the quality of the genomic DNA samples. For example, in this result, we can see the genomic DNA size over than 100 kb is less than 50%. Some special precious tissues will be fixed and preserved by formaldehyde after solid biopsy. These samples are important for clinical diagnosis and scientific researches. However, the DNA and the RNA usually become fragmented after formaldehyde treatment. So the quality control of extracted nucleic acids from FFPE samples are important. The DNA quality number, DQN number, can provide the integrity index for the FFPE DNA. In this picture, we can see the sample 2, the blue line, is partially degraded compared to the sample 1, leading to the lower DQN value. The RNA from FFPE sample usually severe fragmental compared to FFPE DNA because of the chemical sensitivity. So it is difficult to identify their integrity. DB200 is a new integrity index for the FFPE RNA sample quality control in recent years. DB200 is presenting the percentage of the RNA over than 200 NT in the sample. Here are two FFPE RNA samples which are analyzed by QSAP. And we can see the QSAP can provide DB200 value for each sample automatically after analysis. Since 2007, NIH of the United States proposed a global human gut microbiome project. Until today, many scientists involved in the human gut microbiome researches, and more evidence proves that the relationship between gut microbiome and the disease, for example, coronary disease and cancer, even the behavior disorders such as the ASD of children. This is an example workflow for gut microbiome analysis. Scientists extract the total DNA from human stools for next-generation sequencing and then identify the bacteria species by bioinformatic analysis. The extracted stool DNA is analyzed by QSAP platform. QSAP can provide the accurate size distribution for different stool DNA samples. In addition to DNA, total RNA is an important material in biological laboratories. Traditionally, people will use the ratio of 20As and 18As to evaluate the total RNA quality. Now, many people use the ring number as a reference for the RNA integrity index. QSAP can provide the ratio of the 20As and 18As, as well as the auto-assign the RNA quality number, RQN number, for users after analysis. In the right picture, we can see the RQN number decrease when the degraded level of RNA increased. In addition to above applications, QSAP can be applied for multiple applications for DNA, RNA, and protein molecules. For instance, the NGS library QC, SNP and RFLP analysis, mRNA QC, and the protein profiling and the purity check. BiOptic provides 3 QSAP systems to be adapted for different sample throughput: QSAP 1, QSAP 100, and the QSAP 400. QSAP is easy to operate. 3 steps to start the analysis and get the result in 3 minutes. And one detection range and the various cartridges to be adapted for various applications. The detection range can up to the 165 kilobase. Also, the QSAP biofragment analyzers can qualify and quantify nucleic acid samples in single analysis and generated a digital report automatically. In conclusion, the QSAP biofragment analyzers are an automatic quality control system for various samples. Today, Alex and I present an automatic isolation and quality control tools as a total solution for gene diagnosis. iCatcher from the CatchGene and the QSAP from BiOptic can facilitate the isolated and QC workflow and efficiency for your samples. Thank you.